All right, I'm very pleased to introduce the final panel. We've tried to have a special unveiling every day. Yesterday was macro, nano Gigapan unveiling. And today is time-lapse gigapixel imagery unveiling. Before we do that, two announcements. One is that the company Gigapixel Systems, Gigapan Systems, is going to host a cocktail reception at 6:30 at the Holiday Inn in Oakland. So everybody's welcome to come to that, and we once again thank our corporate sponsors at Gigapan Systems for hosting the cocktail reception. The second is many of you have asked our staff how we can share your emails so that we can have some kind of community here. We'll attempt to create a forum entry regarding this group. Most importantly, though, I will talk to my staff, and barring those of you who don't want your email shared, we'll be sure to introduce you all to one another via email, so that we have an email list that you all have access to as registrants for the conference. That is the end of the announcement side. The panel that we're now going to have is entitled Gigapixel Timelapse Panel. Gigapixel Timelapse Systems for Quantifying Phenology from Ecosystem to Organism, and Timelapse Gigapan. So it's several presentations. I'm going to introduce all the speakers first, and then they will speak in turn. The first speaker is going to be Dr. Tim Brown. He co-founded TimeScience in 2006 to develop innovative imaging and time-lapse technologies for scientific and educational applications. He's been collaborating with the Borovitz Lab to develop a solar-powered wireless gigapixel time-lapse imaging system. Many of you have seen the Borovitz Lab images online. The Gigapixel system records 360-degree gigapixel images every hour, and it can automatically upload them via 802.11 or cellular connection for automated stitching on a remote server. And they've developed an online browser for playing back through time these gigapixel images, and you'll see that. Our second speaker will be Randy Sargent, who is also co-chair of this conference. Randy is a project scientist for the Global Connection Project and co-PI of the Global Connection Project at Carnegie Mellon University. He's also a computer scientist at NASA Ames, working on prototype technologies for future Mars missions, and he's co-PI of Gigapan as a whole. Our third speaker will be Janet Stephen in the corner. And Janet Stephen is an assistant professor at Sweetbriar College in Virginia. She's a plant evolutionary ecologist, and she's a botanist. And she's an example of the ideal user for gigapixel time-lapse systems. All right, Tim Brown, welcome.
Alrighty. Can you guys hear me?
Yeah, bring it down.
Good, like that. So our project was basically we set out to figure out a way to do time-lapse gigapixel photography for long time scales, and what my real interest in is how you record from landscapes essentially forever. So you put a camera in there, and how you can have a data stream coming out of that that's scientifically usable for as long a time period as possible. And in the Gigapan context, what this lets you do is actually track every plant in an ecosystem, but across the scale of an entire landscape. And so it's really an amazing change in scale for what people normally do who do landscape ecology. And I think this picture captures really well why it's so important that we do a good job of understanding how ecosystems work. And this is a map you probably all have seen of the United States at night. And it's pretty easy to see from this the extent of human impact on the environment and the landscape. And to me, that really conveys why we need to understand how ecosystems work and how we can manage them better and how we can rebuild them when we want to do restoration work. But to do that, you need to know a lot of stuff, and a lot of it's really detailed and a lot of it's hard to get. So you have to understand plants and animals and their abundance and behavior, population structure and genetics, microclimates, water, and then all the human impacts as well. And these all come together to make the ecosystems we have and the ecosystems we'll have in the future, and if we're trying to restore them, we have to take all these things into account. And Justin Borovitz came up, who's the person I work for, came up with this idea of a GenBank for ecological data. GenBank is great. Someone does some sequencing work, they throw the sequence into GenBank, and if you want to do science on it, you get it back out. And nothing really exists like this for ecological data. So we all do these long projects, we have all these data sets, but it's very rare that they're easily available to others. And it's incredibly rare that you could say, I want to know all of the places where this species of tree has been studied for this many years, and there's images and genetics and quantitative sensor data. we don't have the ability to search for that now on Google. And that's the sort of thing that if we have that, we'll be able to do stuff at the scale that people are doing things with genetics these days. And I think a good way of thinking about that is that every spot on Earth has a biological history. And it could have a data stream associated with it. And you have things like the species that are there and the genotypes and phenotypes. And you also have abiotic measurements, and then you have imagery, which is what I'm focused on and I think is really important. But you need a lot of stuff tacked onto that to make this useful. So if you're sort of imagining the perfect system, I mean, we kind of have it in Google Earth in that you can zoom into any spot on the planet and see pictures. So the field site I'm going to be talking about is in a dune ecosystem. This is just at the tip of Lake Michigan there in Indiana. This is the Indiana Dune State Park. And it's a pretty cool place because when you go down there, you're on a white sand beach. You feel like you're in Cape Cod or the Caribbean, except there's steel mills on either end. And when I was there in the summer, it was like 100 degrees. There were waves. I was body surfing. So it was this beautiful white sand beach, and then there were these steel mills just like belching toxins into the air and falling into the water. So it's a pretty cool place to work. And the site we study is a bunch of these dune blowouts. And so you can imagine that if you're a scientist and you want to do some research, or you want to give kids a research project, they'd want to be able to zoom in like this. And then ideally, you just click right on a plant, and then you just see a time lapse of the plant. And ideally, that time lapse would be 10 years long, however long you'd have your sensors in there. You'd actually have high-resolution image data from every single plant in that landscape that you could just click on. And even better, it'd be great to be able to pop up like all the other microclimate sensor data you have. And even better than that would be able to pop up the genes, from every plant in this little community, or zooming out to every tree in the whole landscape. And that's kind of what I'm thinking about when we talk about this idea of gigavision is, all data all the time from every spot that anyone ever takes on. And to do that, you need data in a standardized format. The way, like the Weather Service produces all these data streams from around the country. They're in a standardized format, and it's really easy for anyone to grab that and do stuff with it. And you have to connect observations and sensor data with a specific place and the species that are there. But to do that, you need centralized repositories and qualitative and quantitative improved monitoring tools. And I think one of the things this will do is give us really much better educational tools, because it gives you the ability to do all the cool stuff everyone's talking about here with gigapans, and take your classes and ask them real scientific questions, and they can go to a data set from nearby or from the other side of the world and start looking at real data that scientists do, using tools that scientists use to ask interesting questions. And so they actually have the experience of doing what feels like science to them, and maybe even really is useful science. And there's a lot of data streams that could go into something like this. So there's all the kind of data we normally collect, like on-site cameras, gigapixel imaging, time-lapse or repeat photography, and sensor streams. But then there's this whole new range of stuff that people can get because we have geotagged cameras and things now. So researchers can come and take pictures or occasional measures. There are all sorts of citizen science apps where people could just take a picture, and that could, it knows what trees you're looking at, and it goes up onto the web as another picture of that tree. And then there's all this awesome stuff with chance photography. So I was looking at Flickr. 4 million geotagged images go up onto Flickr every month. And I found this great graph that said that 0.8% of the world has been geotagged so far with a photo. But that gives you the sense that we're just at the beginning of this, where pretty soon, everyone's phone is just going to be sending out pictures, and there's all sorts of weird things about that. But from a scientific perspective, every one of those is a record of something that might have a tree in the background, or a plant, or a bird. And you can imagine when back-end processing gets a lot better, that you could start pulling real science out of that. 3 images pointed at the same tree, that becomes a data point. But it's not that clear how we do all that. So the typical ways you do this are you might use satellite sensing, and all the stuff from Google Earth comes from these satellites that fly over the Earth a couple times a day. And they get really good data, but it's infrequent, and it's not very high resolution. And also, places where there's a lot of bad weather, like I work in the desert in Utah, so we don't have that problem, but, like Michigan, it's often cloudy. And then there's land-based sampling that we all do. We go out and do field work, and we collect stuff, but it's pretty small scale. It's hard to collect land-based data over a large geographic scale. And so you get this question of how do we scale between those things? And there's been this idea around recently of near remote sensing, which is sort of things like UIVs and kite photography and gigapixel that lets you get that kind of intermediate scale between looking at a few trees on the ground and looking from the satellite. So you can use towers or aerial surveys, both of which have a lot of limits. Or you can use gigapixel imaging, and you can just take a picture of every plant in your entire landscape, and there you have a nice high-resolution photo of it. So traditional phenology being the study of life cycle processes in birds and animals in the environment, it's pretty good in terms of getting, infrequent data sets, but the sample size is limited in time and space because someone actually has to go out there and collect the data. And it's typically non-quantitative. Thoreau took really great records of when trees bloomed and leafed out, but how you convert that into numbers that you can actually do real science on is a really difficult question. And also it's difficult to sustain over a long time because, Thoreau moves away from Walden Pond and there's no one else there to keep taking notes for him. So, it works well, but it needs to move into the realm of modern science where we're applying technology to pull out really, complex and rich data sets. And also irregular events, like if you look through our Gigapixel Timelapse, you can actually find a deer in one frame. And I found it once and I haven't been able to find it again, but there's like a deer there in one day. And you're like, yes. But, stuff like that only happens really infrequently, so unless you're recording all the time, it's easy to miss. And then you have, on the other side, you have traditional lab phenotyping, where you're looking at the genetic structure, the genetic environment that plants live in that helps determine what they look like when they grow up in a certain environment, and that helps you piece out the genetic impact. But it's limited in scope because you can only do it in a greenhouse, and it's difficult to extrapolate that to the field often. And so what Justin's real interest was in funding this project was how do you start taking some of these processes of lab genomics, where we're looking at the genetics of a lot of plants and piecing out the genetic and the environmental component, and bring that into a field situation where we're looking at a real ecosystem that's a lot messier and has a lot more inputs. So to do something like this, you'd like to have plant-level observations for every plant in the landscape and a long-term visual record. And the power of the visual record is really, I think it shouldn't be underestimated, because if you can go back in time, you can do totally new things when you have new ideas. So if an invasive species appears, like in Salt Lake City, there's this new invasive that over the past few years is just suddenly now it's everywhere. If you had a Gigapixel time-lapse of that, you could actually go back to the few places where that plant started growing out of, wherever it got first deposited, and watch the process of how that goes up over the landscape. And you can't do that unless you've actually been filming it for a long time. And then the other way it's really important is ephemeral plants. So, if you're going into the field once a week, you can take a picture of a tree, but that little plant that's going to pop up and have the bright yellow flower, it's really hard to tell where that is in the landscape until it's actually bloomed. And then you can't go back in time and see the previous stages to when you identified it. And also, sequencing now, it's like $10 a plant, but, it's going to be free or 50 cents or something per plant, and, pick your time scale, 10 years. So if you've been filming for 10 years, 10 years from now, you can sequence every plant in the landscape with a $1,000 grant, and then you have that data, but you can go back and apply it for 10 years of field data you've already collected. So having pictures is a really powerful tool for that as well. Ideally, in the future, we'd be able to put in infrared cameras that are doing gigapixel photography, and then, there's a lot of really cool wireless sensor systems. So you imagine being able to just put out little $5 temperature and humidity sensors over the entire landscape, so you really get this like three-dimensional map of what the microclimate looks like in your environment. But it's not that easy to do. So what we wanted to do was to be able to record gigapixel scale images about 4 times a day. It has to survive a huge temperature range. In southern Utah, it's going to be 115 for a month straight. In Chicago, in the dunes, it's minus 10 Fahrenheit for another month on the other end. So you don't want your hard drives to break and everything. It's a solar-powered system, so it has to be really low-powered. We don't want to go out there that often because it's expensive and hard to get to. And because I'm a biologist and I don't have any formal training in anything other than biology, and I like this whole do-it-yourself movement, can a biologist who doesn't know anything about computers build a gigapixel time-lapse unit? And the answer is yes, with enough suffering. But so, a lot of the stuff you'll see in here is stuff that could be done a lot better, but if you want to just use no microcontrollers and cheap PCs, you'll end up doing it the way I did it. So, and we want it to be low cost and easy to use so that it can be serviced by undergraduates with no tech background. And one of the real goals we wanted was it be almost live, so that the picture is that we wanted to shoot a Gigapan and to be able to see it on your desk, an hour or two later. But there's a lot of technical challenges. You have to upload the images, but you only have a cell network often, or something even worse than that. So we upload thumbnails, and then a student collects the full-size hard drives. But right now we have this mess of I don't know how many terabytes of data in different locations, and the hard drive gets full, so you put it on the other hard drive, and then some student takes that and copies half of it, and then the system crashes. So you're just managing all that. The back end of the camera, once you've built that, is this huge data flow that really is much more important than the hardware, because someone's going to figure the hardware out, and it's not that hard. But once you have those images, how you turn them into data is really, I think, the most challenging question. Then we wanted to automate the stitching process and automate file sync and deletion. So ideally, you just walk out there, grab the hard drive, plug it into your server at home, and go away, and the next day, your computer would delete all its files that had already been backed up, and the server would sync them, and they'd be online. And we're almost there. This is a time lapse. This is one of the panoramas in Salt Lake City. We have a camera it's using an internet IP camera, and it takes 700 pictures. But you can see that the stitcher is not that consistent, and the software I wrote isn't that consistent in how it points to servos. So there's a lot of artifacts, and as you zoom into those and you try and layer them day to day, you end up with a lot of headache. And then you need to visualize the data. So we wanted a web-based browser like the Gigapan site, but with time lapse, and that means that your panorama has to have an exact field of view and resolution between changes and all these things. So your camera is going to change, your equipment's going to fail, your hardware is going to reset, and and you imagine over 10 years you're going to be upgrading your system continually. So through all of that, you need to know when I go to look at that one tree that one tree is right in one spot and not wherever it was in the picture that was taken of it. So we have to do automatic offset detection, and just the actual data collection is also a really big challenge. And we also want to be able to co-visualize other data. So, this idea of being able to see all your data at once, that means you want to be able to look at the sensor data and figure out when that big storm made all the leaves fall off the trees. Because that's the real scientific information there, is how what's happening in the environment is connected to on the ground with the photos you're taking. So we built a camera. It shoots a resolution about 1 pixel per centimeter over the 7-hectare area. We actually have 3 of these, but I'm mainly focusing on the one that we've done most of the data collection on. This is about 500 million times the resolution of MODIS satellite. And you can really actually look at every single individual in the picture. And we're working to find tools to map that onto Google Earth and to figure out how to pull real phenological data out of it. The camera is solar powered. It has cellular. This one is at the University of Utah, and this actually has AC. And plugs into the wall, which is really nice because it's on 24 hours a day. But the system's pretty low power, and it works pretty well. This is a hardware slide, so when you download this later, you can pore over this and pick out all the pieces. That's one of the units in the field in the dunes. This one's kind of cool. It's actually up close, so it just shoots. The farthest away is about 30 feet, and it shoots in almost a 360-degree panorama. We're looking at plants that are about this big, that are really rare, that are just kind of interspersed in the grasses there. 5 minutes? Oh, all right, I'll speed up. So this picture inside of the box. Here's a couple of the other units. I'll get to the fun part then. So data, this is the part you all want to see anyway. Let's see. I was just going to show you first here. This is the raw images without any of the offsets fixed. And you can see that there's a lot of variation. So if you imagine trying to zoom in and look at one plant, it's going to be difficult because there's a lot of variation. And the way this works, it always loads 20 to 30 images depending on what we set. So all these little dots are other images that are not being shown. And as you zoom into quicker and quicker, shorter timelines, you'll get more and more of the pictures from that time period. And that helps manage the data flow. Here's where it's aligned. And one of the things to notice is how much variation in the color. So you'll see, notice how much variation there is in the color of the grass, and that really tells you how much variation there is in temperature and water availability for plants in the landscape, which really impacts the phenology of the small plants there. So that's where the offsets are all fixed up and the zoom level is. So you can see that even though the picture's moving around, the actual locations in there are pretty consistent. And we can zoom in again and look. This is zoomed in, and again, notice you can see how it greens up in the middle there, but on the front side it dries out really fast. And this is important because if you think about where you're collecting data, if you collect data from plants in here, they're going to have a totally different life cycle than the plants in there, even though they may be the same species. And that's really biologically interesting, but it means you really have to be careful in which data you collect, because if you collect a few plants there and there and you put them together as one data stream, it's not really telling a very accurate picture of the environment. And so here's our data collection tool, and so each one of these squares here is a plant that we flagged, and as you zoom over here, you can see I don't know if you can see it changing colors. Let's pick like a tree here. So that one, we can double click on here and zoom into it. And you can see that it moves around a little bit, but it's pretty solid. Compared to how this looked like a month ago, it's pretty awesome. And that was the thing. We were collecting data for almost an entire year before we could even look at any of it, because it was only about 3 weeks ago that we got the offsets working, so we could see that it was actually working. And it was pretty sweet. So there's the whole season going by. And I'll just do one more here with so these little yellow flowers. You can see one problem. I mean, just physically showing all this data takes so much screen space that it's just things like that become a hassle to handle. And down here you can change your time period. So right now, see, we're looking at between April and October. And the way this works is you can squish this down so that if you want to just zoom in to like April through June, now we're zoomed in. And you can also drag, so this lets you slide within the time period you're exploring, and then the bottom one lets you drag around within the whole timeline. And I like this one a lot because you can actually see, give it a minute. You can actually, I mean, these plants are about this tall, and that's about a quarter of a mile away, I think, from the camera. And you really can see their life cycle there. And that's a really good example of a plant that, if you go up to the beginning of April here, there's no way you could know that a plant was going to pop out of there. So the only way you could collect data from this is to find it in the middle of the season when it's nice and bright, and then go back and pull out the phenostages. And you can see over here is our data collection setup. So if you have a new plant, you just go, create bookmark, you just draw a little square around it, and then you could go to every time point, so you just go through here until it's bloomed, and then you click on flower open. But you see really fast that this whole concept of phenostages, like the flowers are open, like where are the flowers open in this picture? And where are the leaves out on that tree? It's like a month-long period in which that happens. And so you realize that even the scale of how we collect data out of this has to change, because we have to start thinking in terms of these continuums, which is really how living things work. And this is the most, I just thought this was kind of, this is from November 8th, which I think the camera seems to have gone offline because this is the last picture we got. And there's also stitching issues, so I think the stitcher's been having a hard time because the seasons have changed, and so this is really different from the earlier pictures, and the weather is coming in. So, but anyway, this is right from Monday. This is the last picture that we got. This came in over cellular. And the resolution's pretty good. I mean, considering this came off a crappy cell connection, you can zoom in pretty far. And for doing a lot of research, you could probably get away with even just this resolution. So let's finish this up real quick here. And you can collect data. So here's some phenostage data, and this is the black oaks and the cottonwoods. I think there's about 10 each of these. And the red is flower buds opening. You can see this is basically percentage of trees that have reached the stage of having their flowers open. So starting out, there's none, and over here, they've all got to that stage. And you can see that there's definitely some variation within population. This is where the leaves are opening, and then at the other end, here's where the leaves are dropping and changing. But what's kind of cool is if you look, this is the yellow flowers that we were looking at, the hoary puccoon, and you can see that their whole life cycle takes place in the beginning of the season, and where the drop-off is, this is where they lose their leaves. It all happens in the first half of the season. And in looking at these, there's a lot of variation within the places that are green and the places, that are dry. First of all, a huge thanks to Justin Borovitz, who trusted me at showing up as a biologist who said I knew something about cameras and kept throwing money at me until we got this working. And it's really been a great collaboration, and we've really done, I think, some great stuff. You can find out all those time lapses are available on the Gigavision site. And our website for my company is TimeScience, so if you want to find out more about how these systems work or help fund us to do more of it, that's where you go. And then, of course, on Gigapan, we put a lot of the stuff up there. So if you search under Justin Borovitz or TimeScience, you can find our time lapses. And then these are all the tireless volunteers that spent 15 hours out in the snow while I was home in my office on the cell phone, like wiring things together to get the cameras to work. Thanks. Should we do questions at the end?
Do some questions while I'm setting up, maybe?
Okay. Should we do some quick questions while setting up?
While Wendy's setting up, we can take a couple questions. Go ahead. About how much is the equipment cost overall?
The hardware is about $6,000, but then you have to assemble it, and it's again, it's the back end because you need to assemble it, but then you need to set up your servers so that all the data can go somewhere. You need to step the stitcher, and there need to be people around who have enough understanding of how it works to set. So the way the offsets work, you put in control images. So if there's a camera change or something, you take a new Gigapan and you say this is the current one that you're going to line up to from here forward, and you have to do those offsets manually for now, and then from then on they're automatically done off of that one. So there's a lot of time that I think it'd be about $15,000 to set one up. So the hardware costs are about a third of that.
So hardware's a third of that, the rest is staffing.
Yeah.
Even time.
Yeah, and you'd have to fly one of us out to tell you how to work it, that kind of thing. It's just all expensive.
Citizen volunteerism in science, though.
Yeah.
Right. Okay, let's stay.
I'm really, truly excited to be here at the conference. All the ideas from all the people I've been talking with, all the presentations. It's like head's about to explode here. So I'm up here to talk a little bit about a system that's very similar to the one that Tim has presented. It seems like it's an idea whose time has come. I'm fortunate to be part of a fairly large team of people, and I want to mention that there were a lot of people who contributed to this. Gigapixel imaging in time. I feel strongly that the idea of being able to go and explore a gigapixel panorama is a lot like being teleported someplace. It's a lot like being someplace else. What does it mean to be exploring a gigapixel image of an old image, an old place? Is that like going back in time? Does it let you go back in time and answer questions? So this is a lot like the things that Tim was showing us earlier. I think it really helped to motivate why it is you would want to do this. I think you guys already get that. Some of the ideas leading up to this. X-Res led a group to Gigapan all of the walls of the valleys of Yosemite, and back in 2008, and we had a wonderful presentation yesterday about this, and the idea that after the pictures were taken, there were rock falls, and you could go back in time. You could go back to see the image of the cliff face before the rock fell. It's very easy to get the after picture. It's hard to get the before picture. So Gigapan lets you get everything so you can get the before picture. Another idea kind of leading up to this was a side-by-side viewing. And Ron Schott, who presented earlier today, was the first person who showed me this idea of showing two Gigapans side by side. Are you in two places at once? What is this? What sort of things does this let you do? And Jason Buckheim presented with Ron a really great website for doing this sort of thing. So I just want to show you what it is to be like to be in the past and in the present at the same time. So let's take a look at, this is a rangeland in, I believe, Arizona, that's been, you see the before on the right and the after on the left. These are spaced by about a year. And the difference between the two, there's a little bit of difference in the season, but the primary difference is that the second one the graze, the cattle have been taken out, so there's no grazing going on. So let's go zoom in here and take a look. And you'll see these two panoramas have been registered. Let's see if I can properly zoom here. I don't know how that is doing. Let me refresh here. There we go. So you see we can zoom in a lot of detail and see the difference in the grasses. Let's go look at some grass over here. You'll see the same rocks, the same things in both images. But it's really great because it's very hard to go back in time and ask these questions if you don't have this sort of a record. So the idea of the time-lapse Gigapan or the Gigapan time machine is simply to record periodically and be able to then go explore in space and time. And at that point, you've essentially built a time machine with images at least. Back in 2004, we built a little machine on the back of my house. It's one of the very first Gigapan devices, and it hooked up to a computer and would take backyard panoramas once an hour for about a 6-month period. It was hard to operate. It was very unreliable. We weren't ready to manage the data. We weren't ready to stitch the images, so we kind of dropped the project. But then more recently, the Epic Pro came out, and we realized with a very simple firmware change, we could turn it into a time-lapse capturing machine. And while it's not weatherized in the same way that Tim's nice device is, it was enough to get us going and starting to capture data. So it led us to focus on what do you do with all these images when they came in. You know, Tim was mentioning it can be a challenge to deal with many terabytes of imagery. Well, luckily we have a person, Paul Dill, who wrote a really nice manager for these images coming in that let you understand, you know, did you miss pictures? Are you missing panoramas? Is the camera actually working? Is the lens cap still on? That sort of thing. One of the tricky things about time-lapse stitching, as you're aware, the standard stitching approach to gigapixel images is to take a mosaic of images and look for optical features in the overlapping regions. So you're always shooting these pictures that overlap in space. Well, the time-lapse overlaps in time. So what you want to do is you want to actually look for optical features over time that let you line up today's panorama with yesterday's panorama, or this moment's panorama with the one you took 10 minutes ago. It's a little bit of a tricky problem because some of the things out in the world are staying still and some of them are moving. You know, if you're looking at a plant, parts of the plant are growing, parts of the ground are staying still. So we needed to be able to sort between which is which. So in the process of this, we added a feature that's in the current beta test stitcher that lets you take kind of a prototype panorama and align a new panorama against it. And in addition to lining up the panorama, it will crop to the same bounds. So if you have, it might be time-lapse, it might be something else. Earlier we had a presentation of putting marks out on an archaeological site and then taking them off, and this would be also a way to line up to before and after panorama like that as well. One of the tricky things about shooting Gigapan in general is the problem of setting exposure. And for those of you who have done it, you know that going out into the field and selecting a fixed exposure can be a challenge. Well, there's no photographer out there to select the exposure every hour of the day or every day of the month. So we decided we really needed to solve the problem of correcting the exposure in the stitcher. So we run all of our time-lapse systems with automatic exposure and then correct for that in the stitcher. And I'll show you an image of kind of before after. This is a carnival at CMU. There's a tent, and you can see over on the left there's this funny little spot which is all dark. And what's happened is that the camera, when it took that piece of the Gigapan, this is showing a stitched 5x5 grid, it decided to change the exposure significantly compared to the ones around it. So in the right, we've corrected for that. Once you've stitched all these panoramas, and you might have 100, you might have 1,000 in one of these sequences, you have to have some way to go view and explore that. And we've been working on a new technology for that uses HTML5 based video tags, and it actually inside a web browser will let you go and very interactively pan and tilt and zoom in the same way you can on the Gigapan site, but then also press play and scrub back and forth through time. So we hope to make the time axis just as explorable as the three axes of space. A little bit of technical detail behind that. The Gigapan viewer, to show images as you zoom in, divides your image into a multiple resolution set of tiles. So there are coarse tiles when you're zoomed out and fine tiles when you zoom in. As you zoom in, it just replaces the coarse tiles with the fine tiles. For the time-lapse viewer, we replaced the tiles, the image tiles, with the video tiles. So as a result, just like the standard Gigapan viewer is showing you maybe 20 little tiles or 30 little tiles all at the same time concatenated together on the screen, this viewer is putting 20 or 30 videos next to each other on the screen. There's a lot of videos for a web browser. As a consequence, it tends to crash the web browsers just a bit. So what I'm going to do is I'm going to show you some of the videos that we've created, some of the time machine instances, if you will, things you can go into and explore. And at the moment, these demonstrations I'm going to show you tend to crash the web browser in about 2 or 3 minutes. So I've actually pre-recorded this. So these are web browsers that I'm going to press play on a video to show you what I was able to record earlier. So this is think of this as a preview of what will be on the web hopefully soon once we work out the crashes with the web browser. So this is the same carnival location where you saw the tent, and this is before any of the building started. We're taking Gigapans every 10 minutes for about a 10-day period. There's about a 6 or 7 day setup, a few days of the carnival operation, and then a day of teardown. So let's go ahead and start that up. So you'll see everything getting built. And we can zoom as we're playing. As we're panning around and zooming, new videos are being brought in and old ones are being dropped off the side. So now everything gets torn down. And the next day, you can see it's a parking lot. So let's go back to the beginning here. And now let's go a little bit. Let's go explore a bit. So I'm just going to grab the slider here and move to somewhere in the middle, and let's look at all the carnival booths as they've been built, and let's pick one and see how it was built. So we'll zoom into this one over here with the towers. And we'll go backwards in time and see it being unbuilt. And we'll click on play and watch it being built. So let me switch to another data set. This is an outdoor garden very close to where you guys had where we had the keynote this morning, in fact. This was taken from March until the present day, so you'll see some of the trees grow leaves and then lose them again. So let me play first kind of from the zoomed out. It's a little bit boring, and you can kind of see things happening if you look really closely, but not so interesting until we actually go zoom in. So let's go back through time and take a look somewhere else. So as we go forward through time a little bit, at the beginning you'll see some of the trees grow their leaves. Let's go in and see one of the trees that was flowering. Let's go back and see it flower. I'm just grabbing the time slider and going back and forth here. Now let's go to the end where the trees start to lose their leaves. You can see over on the right there's a tree that turned red. So we'll zoom in there. Let's take a closer look at what's going on. This is just grabbing a time slider and going back and forth here. There's one other piece here that as you go exploring, you'll notice there's a little slope down below the sidewalk, and it's a fairly steep slope. And this is what it looked like at the beginning. And as we go forward in time, you'll see, poof, something changed. Let's go back. So the whole covering got lost in the middle of a big storm. And this is one of those cases, like Tim said, where you really want to connect this to weather data. You want to see the weather, as you go forward. So as we go forward through time, you'll see some of the weeds start to grow, and then boom, they get lost again. So we go back and forth here. So this slope has been a constant issue maintaining this garden. Yep, there they go again. And let's go all the way to the end, and you'll see our new attempts to revegetate that. Put lots of sprinklers and some new surface material. So it turns out that once we built the viewer for looking at gigapixel scale video, things coming from GigaPan weren't the only data sets. This is from a physicist here at CMU who does simulations of the early universe, and he basically builds these huge, multi-billion pixel simulations of gas condensing and turning into galaxy clusters and galaxies and things like that. So let's go play with this data set. So I'm just going to grab the time slider and move slowly through here. You'll see everything's kind of coalescing into these bands, and that's where all the action's happening. That's where the galaxy clusters and stars are being formed. Go backwards, and let's zoom in and see in a little more detail what's going on. So these videos will be up on timelapse.gigapan.org, and as soon as we get the we can keep the browsers from crashing, the whole experience will be up there. I'd like to give some credit to a number of people who've been beta testing the system and actually going out and taking the pictures. At the carnival, we had people going on the roof of a very tall building and some cold weather to replace cards every day in the cameras, and I just want to acknowledge everyone. One person in particular, Janet Stephen, has been beta testing, and she came up with a particularly compelling environment that she'd like to share with you.
Before I start, I have to thank Randy and Ela and MJ and everybody for making this such an incredible experience. I've had such a good time just being so creative and thinking broadly and being interested in lots of things that aren't plants, and it's just been spectacular. But now I get to tell you about plants, which are one of my favorite things, next to evolution. And one of the things I like about plants is that they are so interesting. They're dynamic and they respond to their environment and they change all the time. But they do it in a time scale that we're not accustomed to. They do it in their plant time scale. So in order for us to really understand plants, we have to speed them up so we can understand them. And time-lapse has been used for a long time to understand plant behavior better. And the beauty of Gigapan time-lapse is that we get plants at the population level and plants at the individual level. So you can watch what all the plants are doing together, and you can watch what individual plants are doing. So before I hit play, I want to tell you a little bit about how I set this up. I used a species of plant that's very commonly used in classrooms. This is actually a strain of Brassica rapa that's been bred specifically for classroom use, and it's called Wisconsin fast plants in the classroom. And so my growing setup is what you order from Carolina Biological Supply to grow your plants in the lab. So I just set up a bunch of Brassica. I used the dark room because I wanted to keep light, ambient daylight from affecting what was going on, so there's just one constant 24-hour fluorescent light. I think it's kind of ironic that our old dead dark room is seeing new life in, imaging, but in a different way. So I have 2 cinder blocks in the picture because Randy told me to put them there. And he's telling me, I'm assuming he's not playing a practical joke, that they're for having something static when you stitch, so that because the plants change so much, there needs to be something that's not changing. I used an Epic Pro with the time-lapse function. I took a 7x3 pano every 15 minutes, and I set this up and ran it for about 4 weeks. I'm not sure exactly how many, but so I ended up with about 250 gigs of pictures and all, and I mailed them to Randy, and he made them, these videos that you see. These are also pre-recordings, so this is a recording of somebody using the browser. And I think there we go. So there's a couple of things to watch out for. You should watch to see them flower and watch flowers open and fade. And also, I put caterpillar eggs in with the plants towards the end of the video. There's a species of caterpillar that's specific on Brassica. And so there's a few points at which you can see caterpillars and you can see caterpillar eggs. I think at the very end here, there's a caterpillar that's going to make an appearance. Oh, there it is. You see him in there? Right there? So we can back up and watch him in action again. Okay, so now we have another video, and there we go. We'll watch this one. And I don't know if we can focus. Yeah, there we go. That right there is the little piece of wax paper that has the eggs on it, the caterpillar eggs on it. So the caterpillars hatch from there, and they move the paper around, which sort of surprised me. And you can see the holes start to appear in the leaf. And then the leaf just crinkles up. In case you missed it, we'll back up. There's a little spot right there where there used to be a flower. and then it gets eaten. And I think there's one more video in here. Is that right? Oh, it's still going. So now this one is focusing on one plant that was behaving very strangely, and I'm not sure why this plant was doing what it did, but it was doing fine, and then it fell over, and it never quite managed to straighten back up. So you can see it over there kind of flipping around. I'm not really sure what that was about. Yeah. So we can zoom in more and say, what's going on? Is it caught on something? Is it hitting another plant? And it looks like it fell over and somehow didn't get the stability to get growing straight again for a little while. And finally it straightens back up. So when I originally started this project, I imagined that I would use it a lot in teaching, and that it would be an excellent opportunity to look at a population of plants growing and changing before you set up an experiment, a way to make hypotheses, and possibly a way to collect data. But the more I work with it, the more I think about it as a way of capturing data that we can't otherwise see, capturing data that up to this point we couldn't do because you couldn't measure the same thing every 15 minutes for a month straight. So I'm really starting to think about ways in which we can ask questions about plant phenology and plant growth that we couldn't before because we have this fine scale data set on so many different plants. So I consider this a rough draft. Hopefully I'll be able to put together more film soon. And that's all I've got, so I'm happy to take questions.
We got time for a couple of questions for all three panelists. Go ahead, Gene. What did you say was the interval for the lab?
Every 15 minutes? Yeah.
Excellent.
How long does it take to create the final product?
So thanks to Paul, the stickers are a lot faster than it was a year ago. So it's taking like Tim described, we were also capturing data before we could really process it. So processing everything at the end, it was taking several computer days. So if it were on a single computer, it might take a week to do all that. But if you're doing it as the gigapans are taken, a single machine will have no trouble keeping up with a camera.
So you'll be just adding more frames to the movies, so to speak?
Correct, right.
So you can pull over.
I was wondering what you're working on towards annotating and annotation of these series. Displaying of annotation.
Me or?
Don't look at me.
So we haven't really gotten that very far there. I saw some really great stuff that you've done, Tim.
Yeah, and I think I forgot to show, we have the integrated graphing too. Did I show that? Where you can play weather data and there's a line that goes along so you can see the wind event actually in the wind charts along with that. But you see the problem is how you visualize all this data on a normal screen. Like I was back there and I was doing it on my laptop and then I realized this was square and I was doing wide there and I had to reload everything. And it just gets hard to fit it all in there. I mean, you just have to buy a bigger and bigger screen, and then when you want to demo it for somebody, if someone's on a netbook, it's worthless. So I think we really start to have to think about how you actually show that much data, along with the tools to actually collect it.
And this viewer does have a little snap lapse function where you can film a snap lapse of you moving around in the pano, so that might be a good starting point for annotation. Go ahead.
How did you handle the focus? Because there are many instances of that.
So I set it on manual focus and left it, and there are definitely some plants in there that fell further back than I thought they would and ended up out of focus. So that's something I've been thinking about. I tried to set the focal depth as deep as possible.
On ours, the one with the wide view is on manual. The one that's close up is actually on autofocus. And our capture program, if it can't focus, it puts in a black frame, so the Gigapen actually gets built with spare frames in it that were missed images, so it can still stitch.
Yeah, and for all the other ones you saw, the videos of on my presentation, they were all with manual focus, pretty much so at infinity.
One final question. Anybody want this? Yeah.
So, Randy, are you going to restitch the 6-year-old images from your backyard?
And once I find them all, I will stitch them.
That's the end of our panel discussion. Let's thank the panelists again. The dead silence in the room when they were showing videos is very loud. There's a lot of